Hey, biologist, Mr. Fuad here. Today, we're going to be talking into our chapter 6, an introduction to metabolism. We're going to start to get into cellular energies. So, first up are the need to knows. The structure of enzymes includes the active site that specifically interacts with substrate molecules, and for an enzyme mediated chemical reaction to occur, the shape and charge of the substrates must be compatible with the active site of the specific enzyme. So, what enzymes are going to do, they're going to help drive these chemical reactions. So, here you may recall from like standard biology here, or chemistry rather, that here we have the reactants and the products, kind of like a chemical reaction, moving all the way from here's our first reaction, our starting molecule, into one, two, three, final product. What this is kind of getting at is metabolism. Is basically all of an organism's collective chemical reactions that they are going to take place internally here. So the summation of all of these chemical reactions we refer to as a person's metabolism. This in part is going to manage the materials as well as energy resources in a given cell. So obviously different cells are going to do different chemical reactions, and so you might need different sets of things for different types of cells. Liver cells versus heart cells versus kidney cells, and so on and so forth. If we were to take a look at the true metabolism, I believe I may have showed this in class once or twice. This, in reality, is metabolism right here. This is every major chemical reaction that takes place, all of the building of molecules, the breaking down of molecules. We are not going to focus on all of this. This is like into doctor school. So this is why doctors go to school for so long. You could literally spend years and still not even have a true grasp of everything that's going on here. So this is the true graph of what metabolism is all about. All the summation of every single chemical reaction. It's pretty daunting, to be honest. But again, we're not going to focus on that. We're just going to pick little parts for now and tackle it one little thing at a time. So when talking about these chemical reactions, we either have two different types of, we call them pathways, or types of chemical reactions that can take place. We either have catabolic or anabolic. So for catabolic or catabolism, sometimes we kind of summarize it as, this is the releasing of energy by breaking down. That's the key here, breaking down complex molecules into simpler compounds. For instance, if we happen to be digesting some things, so like amylase, it's in your saliva and it breaks down starches. And so it will break the food down to try to release the energy for your body to easily absorb it and process it. Again, jumping back into the whole metabolism chart. Now, anabolic, on the other way, is going to use energy or consume energy. We are building complex molecules from our simpler ones. You may recall way back when our monomers building up to our polymers. This is what we're referring to are these anabolic pathways. For instance, amino acids linking to form different muscle protein. Now, within all of this, there's a few other terms that we do want to mention. So, catalysts, we're not going to focus on today. We will eventually get there. But what an enzyme actually is a biological catalyst. And a catalyst, by definition, this is something that can change the rate of a chemical reaction without actually altering the process itself. And so what it actually does is this definition down below is speeding up this chemical reaction by lowering the activation energy. It lowers this barrier that's needed for a chemical reaction to be driven forward. And we'll talk more about what a catalyst is and how these play out in the next set of notes. But for right now, we're going to focus specifically on enzymes. So sucrase happens to be an enzyme right here. So we have our sucrose plus water right here. Those are the reactants, arrow over here. And usually when we see these, and how do we know that an enzyme is going to be involved? One, it ends in ACE. All enzymes are going to end in ACE. And notice how it is above the chemical reaction arrow. This is a very common thing that is shown in biological types of chemical reactions, is that they'll put the catalyst or the enzyme here above the chemical reaction. So In the products, we are now splitting apart the sucrose into both glucose and fructose. So we took that disaccharide and we made them to monosaccharides. Also notice what we did here is that we split apart the water molecule. We have hydrolysis is actually then taking place as well. So we are splitting apart our water molecule to then break down in these particles. So this right here would be catabolic. So this is a catabolism type of chemical reaction by splitting molecules apart. Now back to enzymes, which again is our focus for today. Reactants in an enzyme, we call these the substrate is what we do. So we use, it's very similar to chemistry and chemical reactions. We just use a few different terms. So in part of the reactants are going to be referred to as substrates. When this substrate is going to bind, it's going to form a loose type of bond, if you will. We call this the enzyme substrate complex. The two of these are going to then come together to form this little complex here. And the specific place at which the active or the enzyme and substrate are going to meet is referred to as the active site. Now here's a quick little GIF just to show you. So the giant yellow blob right here, giant colorful blobs or proteins, is going to be the enzyme. But this little open slot right here, this is the active site that we can see. Here's our active site. And then if we had a substrate, the substrate would come right in. Now notice this active site has a very specific type of shape. And this is key because again, recall, the shape, or sometimes referred to as conformation, is going to determine the function of these proteins. So once again, the shape will determine whether this substrate can bind to this specific protein. And it really is, as we're going to be referring to it often, a lock and key type of mechanism. So here it is, just a little bit more, another good example. So let's say we have our two reactants over here, again, just generically referring to them as substrates. They are going to then find their way to the protein here, and that's a giant purple blob, here's our enzyme rather, and once they bind together, now it is an enzyme substrate complex. These are now going to be held together. And again, it's a very specific shape. So we have this little rounded part on top with the red one, this pointy bit down on the bottom and the green one, and they have to fit. And if they don't, that's a problem. Next, the chemical reaction will take place. And now the reactants, just like in a chemical reaction, are going to become products. Also notice very carefully here, the enzyme, it too kind of rearranged itself. So no longer is it the exact same shape, or rather, the colors of the substrates are going to then subtly change. Afterwards, these products are going to then release. So now these are going to be our products down below. And what's key here is that the enzyme is not used up. So this is not technically part of the chemical reaction, meaning that it's not going to be part of the reactants or the products, which is why in that chemical reaction, we put it above the reaction arrow. It's helping to drive the chemical reaction forward. However, it is not part of the chemical reaction. So something that's very key about our enzymes in these anabolic and catabolic chemical reactions. So here's a few other illustrations just to demonstrate. So here is another example of catabolism, or a catabolic chemical reaction. So notice how we have a very specific shape. Here's our substrate fitting into, here's the active site. This giant gray blob, if you will, is going to be the protein. So now it needs to fit into this. Now the chemical reaction will take place. This is sometimes referred to as the intermediate. It's kind of the in-between. So if this is reactants, this is products, this is intermediate or in-between, and then the products are going to be released. So notice how the reactants were split apart into their two pieces right here. Now, oppositely, if we do the same thing for an anabolic type of chemical reaction, we have our two reactants. Now they're slowly going to piece themselves together. Notice how this first one had to go in for the second one to fit in. And this is true for these anabolic types of chemical reactions, that it must be a very specific shape. So they have to fit into the active site, perfect. Afterwards, here's the chemical reaction taking place. Here's the intermediate into our step 3, and then finally being released as our final product. Part of this, sometimes referred to as an induced fit, and that is where the shape of the enzyme will then slowly, like, kind of give it a nice little hug. Is kind of what it does when it forms this enzyme substrate complex. So here's our substrate. This is more of a molecular type of model right here. Then here's this open space substrate, or the active site here, and they must be specific to this location, whatever the substrate happens to be. Afterwards, here's the intermediate. There's the enzyme substrate complex, and this is where the chemical reaction would then take place. Afterwards, our final step would be the releasing of the final products. So, thank you all for watching. Please let me know if you have any additional questions. Have a fantastic day.